So welcome again, everyone. We're going to kick off the scientific session this morning. The first talk is by David Warburton from the Children's Hospital in Los Angeles. His talk is titled Developmental Context of Rare Lung Diseases and Translational Approaches to Correcting Alveolar Milieu.
Well, thank you very much, and Luca, thank you, and the committee, thank you very much for the invitation. And as a representative of the City of Angels, we get earthquakes. We understand that, and we're very sympathetic to you with that. So, because I actually come from Hollywood, our children's hospital is actually located in Hollywood, California, I thought we'd begin with a game of Hollywood Squares. So, just shout out, who are these people? Who's that? Marlon Brando. Jerry Lewis. Evel Knievel, that's right. And what do these three fine gentlemen have in common? Yeah, lung fibrosis, that's right. They all died of different kinds of lung fibrosis, and here they are towards their end of their lives. Of course, Evel still has the coolest motorcycle of the three. But you invited me to come here as a pediatrician and developmental biologist, and the point I'd like to make is that you doctors that take care of these people think this is a disease of old age. Actually, it's a developmental disease, and I'm going to show you some data on that as we go along. Now, the first thing I'm going to show you, this is the developing lung, and I'm not going to talk about that very much in this talk because we did that in Australia. But I just want to point out the lung is not a simple organ, and in fact, the lung and the cerebral cortex are the two most recent things to be developed in evolution. And so that was exciting. They can both go badly wrong, and my cerebral cortex is not working so well because I just flew from America. But anyway, so the lung is composed of epithelium, mesenchyme, blood vessels, and nerves, to name but a few, and these things have to be exactly correctly organized for it to function. So there's plenty of margin for error in this. Now, that all occurs during early development, and the interesting part of lung development from a clinical point of view is the alveolar phase. And this is the famous Fletcher and Peto curve of lung function. Development of the alveolar surface. Now we think the alveolar surface is probably developing up until at least the age of 20 and maybe later. But past the age of 40, we lose 2 to 3% of our lung function every year. So I'm sort of over here somewhere probably. And eventually, if you produce this curve, the lines cross out here by the exits, appropriately by the exit sign here. So this is what will happen to everybody, that if we cure cancer, heart disease, and Alzheimer's disease, everybody's going to die with a bottle of oxygen because the lung is the final common pathway. So the lung is actually a fairly dumb organ. It has a fairly simple response to trauma, infection, and environmental damage, which causes tissue injury and inflammation, which then, if you're lucky, leads on to repair. And this is mediated by TGF-beta and VEGF and a number of other things. If you get too much TGF-beta, you end up with fibrosis. If you're an adult, if you're a baby, you get too much TGF-beta, you don't make alveoli, and you have a disease called bronchopulmonary dysplasia. Now, it is possible to make IPF actually in fetuses. This is a fetal monkey, and it was injected with one of Jack Goldie's adenoviral TGF-beta1 viruses, and the virus was injected over here. The virus is long dead, but this interstitial fibrosis has proliferated through the tissue and actually reached the pleura and caused this bread and butter pleura. One of the questions in the field that's really unanswered is how does this fibrotic process proliferate through the interstitium of the lung? It may have something to do with mechanical issues, and Jack will talk about that to bookend the end of the conference. Now, I mentioned TGF-beta. This is the TGF-beta pathway. And Wei Shi is sitting in the front row, and he is my senior associate professor who colleague who's focused pretty much on this SMAD3 molecule because there are mice available to study that, and it is a nexus of the canonical TGF-beta signaling pathway. And if you look at these mice, this is the wild-type mouse up here, and you can see that in mice, alveolarization occurs postnatally, which is very convenient. And these holes here in the lung get smaller and smaller and smaller as the alveoli become subdivided. But if you compare that to the SMAD3 knockout, you see the holes start to get smaller, but then they start getting bigger again. And if you plot this out as mean linear intercept here in the purple color, you can see the alveoli form, but not completely, and then they start to fall apart again. And it turns out that at about a month of life, there's a burst of protease activity, which is probably responsible for this degradative phase of alveolar destruction. So the message from these mice is, number one, the alveoli have to be constructed properly. And number two, if they're not constructed properly, then they're susceptible to dissolution. And if you do a double cross of the SMAD3 mutant and the MMP9 mutant, you can rescue this process. So this is a proteolytic process which is in the TGF-beta pathway. Also, not to be ignored, are the immune cells, which are in increased numbers in the bronchoalveolar lavage of these animals. And probably what it is that TGF-beta is actually acquired to suppress the immune, to control the immune response in the lung. And this is probably what's going on in lung injury, but then too much TGF-beta is a bad thing. It's too much of a good thing, I guess. And also, in these mice, there are major problems with the mesenchymal stem cells in the lung. You can see a mesenchymal stem cell colony-forming assay here done by one of Way's people. And there's a significant reduction in colony-forming units in the SMAD3 deficient mice. Now, this is a picture that I got from the back of a travel magazine. And it's dated 1930. It's ethical tobacco advertising from 1930. And it says, 20,679 physicians say Luckies are less irritating. And this beautiful young lady, who you notice is the target of the advertising campaign, nothing changes, does it? I mean, they're still doing this. She says, I too prefer Luckies because they're toasted and they protect your throat. I mean, it's bizarre, isn't it? So in California, we just had a proposition to put $5 extra costs on a packet of fags, and it failed because the tobacco industry used exactly this pitch, that physicians disapprove of this. So they're still doing this, these buggers. I mean, so anyway, so if we thought, well, what happens to these mice when you smoke them? And we thought, well, these SMAD3 knockout mice, why don't we try some secondhand smoke and we pretend that their grandmother smokes or something like that, right? So we put the mice in a smoking machine and they get secondhand smoke from the cigarette. And interestingly, the inside of this chamber looks exactly like a British Railways waiting room in 1954. It's covered with yellow clag, so the second-hand smoke is really bad. Direct smoke doesn't do that, interestingly. But if you do this to these mice, so this is a wild-type mouse, you smoke them for 3 weeks and nothing happens. You do that to a SMAD3 knockout mouse and look what happens. They get accelerated alveolar destruction. So this is an emphysema-like phenotype. And interestingly, these mice are protected against fibrosis. So if you take these SMAD3 mice and give them bleo, they don't get fibrosis. And Way has a poster also showing that if you block TGF-beta signaling in the epithelium of the lung, you also block fibrosis. So it's two sides of the same coin. Fibrosis, emphysema, and SMAD3 appears to be a major pivot. Also, if you knock out TGF-beta type 2 receptor, this time in the lymphoid lineage, you get massive lymph nodes, and you get increased broncho-associated lymphoid tissue, BALT. And if you smoke these mice, they also get massive destruction. So TGF-beta signaling suppressing the immune system probably has something to do with this as well. So here's the sort of idea behind all this, that defective TGF-beta SMAD3 signaling gives you abnormal development, reduced lung capacity, which may have mechanical issues, abnormal immune system, altered inflammatory response. You then go behind the bike sheds and start smoking at the age of 7, and you have cigarette smoke, which disrupts lung homeostasis and leads to alveolar destruction. Now, there's another important principle here, is that these events may be genetic. There's some GWAS evidence that there are defects in the TGF-beta pathway, certainly in emphysema and possibly also in fibrosis. But this is an epigenetic event as well, in that SMAD3 activates this gene P3, and P3 is involved with June, SIRT1, and P65 and P50 in doing this acetylation decoration of the MMP9 promoter and turning it on. In the absence of SMAD3, this transcriptional pathway, epigenetic pathway, doesn't work. The epigenetic decorations don't occur, and MMP9 is off. So if MMP9 is on, locked on, this gives you emphysema. If it's locked off, you can't clean up fibrosis. Because pulmonary fibrosis can be thought of a bit like the Naples garbage strike. Remember the garbage strike in Napoli, where all the garbage piled up in the streets? It wasn't because people were chucking more garbage out of the window. It was because the garbage trucks were on strike. So you can think of pulmonary fibrosis and emphysema as being the flip side of two things, and that this epigenetic switch may be actually extremely important. And it may explain why you can get, and since this is cell autonomous, it may explain why you can get emphysema and fibrosis in the same lung. So you'd have to go down to the single cell level to do that analysis and prove it, but that's the hypothesis we have at the moment. And here's the old Fletcher and Peto curve. This is Roger Bannister up here in the green, or Tenzing Norgay, somebody who can climb to 40,000 feet without getting short of breath, and he lives to be 95. And here is the blue one is a wild-type person who smokes. They get emphysema later in adulthood. Here is a mutant who doesn't smoke, and here is a mutant who smokes. So if we could screen for these things, we could at least tell people, move to the seaside and don't smoke. And that may actually be a very important thing. So what can you do about it? Now, yesterday I went to some of the advocacy talks, and there were a lot of people there that had had lung transplants. And this is what happened with this boy. He's got the other kind of fibrosis, the cystic variety. He's got cystic fibrosis. So he got transplanted at the age of about 14. But I have heard, actually, there are patients on the transplant list, actually, at some of the pediatric hospitals around the world that have IPF at that age. So it is actually a pediatric, in rare cases, it's actually a pediatric disease too. So maybe those, but anyway, so this is replacing the whole schmear. This works great, and those of you who know Julia Pollack, she's had her lungs for 25 years, so when it works, it works. But about half the time it doesn't, and there aren't enough motorcycle accidents to go around. And this boy got a lobe from mum and a lobe from dad. So that was potentially a 300% mortality operation if the surgeon hadn't done it correctly. So actually we stopped doing that. So that's a major issue. So one solution that we've come up with is to use the cells that are found in amniotic fluid. And it turns out that amniotic fluid contains cells floating about in there, and about 2% of these cells are c-KIT positive cells that have stem cell-like features, and this has all been extensively published by our group and other groups. And if you take these cells and inject them into the embryonic lung, shown here as the red dots, you can get them to go into the lung. They will start expressing lung-specific genes, but they don't do it terribly efficiently. As you can see, they're sort of dotted about. You don't see any clones forming. And here's a higher power view of that, some there in the bronchus, around the bronchus, some in the primitive branches. If you do this in vivo, you inject the cells into the tail vein. Because of anatomy, they go straight to the lung, and they actually sit in the lung for a while, and then they come out and start circulating around the body. And this mouse had not been any lung disease. This is actually a control mouse. So it ends up in the liver and the spleen. A few of these cells will persist in the lung. You can find them 7 or 8 months later. And actually, if you go and do autopsies on women who have had sons, you can find Y-positive cells in the body, all over the place, actually. They're rare, but they're there. So this is a physiological mechanism which has been going on for thousands or millions of years. And these cells will, in the face of injury, they will actually incorporate inefficiently and make single cells. So here, that was the alveolar cell. This one is actually, in the naphthalene model, you can get them to do it in the trachea as well, but it's very inefficient. So we thought, well, what else can these cells do? So Orquídea Garcia, who's rather a clever young lady, a graduate student, she said, well, let's try injecting these cells, and maybe they have anti-inflammatory properties, and this might be very useful in fibrosis. So we used our favorite easy model of fibrosis, the milieu model, which of course has nothing to do with IPF, but it's tractable. So here we have the cells are given, the mice are given bleomycin, they're then given AFSCs either at the time of injection or 14 days later, and then everybody's sacrificed at a month. And you can see there's actually a big difference here. This is the standard milieu picture. Here's when you give the AFSCs at day 0, and here's when you give them at day 14. So here you never get fibrosis in the first place, and here you get resolution or arrest of the fibrosis, and this is reflected in the Ashcroft score and hydroxyproline content. So that's great. So then what's going on in the milieu of the lung? So this is at 72 hours, and you can see actually there's increased, in the blue bars, there's actually increases in most of the inflammatory mediators. So early on, there's actually an enhancement of inflammation in the tissue. But if you look at the BAL, things start to get more interesting. And here, we're looking at CCL2 here. CCL2 is driven back towards normal in the presence of the AFSC. And so just in summary for this, CCL2 is decreased, temp is down a bit, complement's up a bit, and so on. But we're going to concentrate on the CCL2 story. So the first thing is that these cells actually concentrate in the fibrotic areas of the lung. So this is red dye attached to the cells in the fibrotic area. So they home to the right place. And what they do there is that they actually affect the milieu within the fibrotic tissue. So this is the cells stained for CCR2. So they make the ligand and they have the receptor, and so in the acute phase they're attracted into the inflammatory fibrotic milieu by the CCL2 mechanism. Here is a migration curve in mouse, and here's a migration curve in human, and they persist in this up to 28 days at the site of the fibrosis, and but at 28 days, in the presence of these cells, the level of CCL2 goes down. So the point is, CCL2 goes up. It's actually made by the alveolar epithelium. It's made by the alveolar epithelium. And at 28 days, it's down again. So the cells then are attracted by the CCL2, but then suppress it. And how does that work? Well, being a very bright young lady, Orquídea ran this gel, and I looked at it and thought, well, there's nothing much there. But she said, well, wait a minute, there's a 0.4 kilodalton band shift here. And it turns out that if that 0.4 kilodalton piece is cleaved off of CCL2, it becomes a dominant negative. So we think this may be an important mechanism, is that the cells are actually making MMPs, particularly MMP9, your favorite, which can cleave this. And we've done this in vitro, and we've done migration assays and everything else to kind of make this point. And what does it do to lung function? Well, these are the PV curves, and you can see the PV curves do improve in the presence of these cells. So we think this is an interesting potential translational approach comes from developmental biology, didn't come from the adult clinic. But translational medicine, well, we always have to be very careful, don't we? We're going to try and put things into patients. I think Thomas Edison put it best. He's the chap who invented the light bulb, and he had more patents than I've got, and he invented something useful, unlike oneself. So he said, we know less than 1/1,000,000th of 1% of what we need to know, and that was in 1900. So I submit that as a result of recent genomic advances, we now know less than 0.001 millionths of 1% because we have a better appreciation of our ignorance than we did in 1911. So, this is the Saban Research Institute where the work is done. And you can tell it's Los Angeles because there's a palm tree in the corner. And the Sabans, do anybody who have children know what the Mighty Morphin Power Rangers are? The Power Rangers, TV characters? Well, Haim Saban made his first $1 billion out of that, and if you bought one of those characters, thank you very much. This is a $50 million building for which they wrote a check. And this is the view out of my office window last week. This is the Hollywood sign. This is a space shuttle coming back to Los Angeles to be located at the Science Museum in Los Angeles, the Space Shuttle Endeavour. Here are some of the people that helped do this, particularly Orchidia. I want to recognize her, and Wei Shi, and Bing Zhu, who did the epigenetics work. And of course, Jack and Martin are in here because they're major collaborators, and Barbara Driscoll, and Laura Perrin, and Roger DiFilippo, and Gianni Carraro, who did the original stem cell work. And here's where the cash comes from. There isn't quite as much from the NIH as there used to be, I have to say, but we still get some. And thank you for your attention. I'd be happy to discuss it.
There's a microphone on one side, and I'm not sure about the other room.
Speak into the microphone.
So, David, I'll start. I wondered if you might be able to tie the stem cell information a little bit more into the beginning of your talk. For instance, are there differences in SMAD3 signaling related to the stem cell treatments, and does that get back to the MMP9? You mentioned that briefly.
Yeah, well, we don't know about SMAD signaling in these cells, but we do know something about the protease environment that they create, and they do secrete MMP9. So if you want a visual image of this, it's sort of like they're sort of like the fire brigade, and they drive up in their truck, and the firemen all get out and they pee on the fire with specific molecules, and it's a complex set of molecules they do this with, but the proteases are very important because the proteases actually modify the endogenous cytokines that have been secreted by the tissue and turn it into a dominant negative situation, which then calms the whole thing down. The cells then eventually undergo apoptosis or swim away and get eaten by the spleen. So they're not going to sit there and make a tumor, and these cells don't make tumors because we've had them in mice for years and they don't. So we think this is actually very interesting. They're also less immunogenic than MSCs, and they work a whole lot better than MSCs in this type of situation.
Can I ask a follow-up?
And I'm corrupt, by the way, so I'm corrupt because we're making a company out of this, I'm just saying. It's for sale. So am I.
Can I ask a follow-up question? You said something very interesting about these Y-positive cells being found in mothers.
Mm.
Would you hypothesize that smoking mothers would have potentially higher Y-positive cells because there's potentially more inflammation and more of a need for these cells to go in? Has anybody thought of looking into that?
No, but that's a very interesting question. Steve and I was saying earlier, I think it's why women are better at multitasking, actually.
And in terms of the receptor system, when these cells appear into the site, often they downregulate their receptors. So what do we know about the sort of receptor repertoire when they get to the site? So how do they get cleared? Because it is presumably they arrive, they download whatever cargo they're carrying and potentially repair the lung or deal with inflammation. What do we know about the clearing them or getting rid of them? Because presumably they don't engraft.
No, I never said engraftment.
No.
You noticed that, right? Yeah. I didn't say that because they don't. Well, they do very inefficiently engraft, but it's not the major thing they do. The major thing they do is fiddle with the environment. If you have no injury present, the cells get stuck in the lung because they're big, and then they kind of squeeze through the pulmonary circulation and go on and circulate and get into the liver and the spleen where they're eaten probably by the Kupffer cells or something. we do know that they get cleared after injury eventually, but we don't really understand how that works. This is kind of early days, so they're very interesting downstream questions.
David, just regarding your CCL2 work, you suggested translational applications, but delivering CCL2 or antagonizing CCL2 could have major implications rather than just specifically on fibrosis.
Well,
so would it require delivery via like a stem cell or specific delivery, or do you think you can apply it more generally?
Yeah, well, I think one of the big challenges in this field is that if you give a very biologically active molecule intravenously, it goes everywhere. And then, of course, you get biologically active side effects everywhere. And so the beauty of the stem cell vehicle is that these cells actually swim to the site of injury, detect what's going on, integrate it using their onboard computer, their genome, right, which is capable of solving billions of calculations at the speed of sound, and then P exactly the right molecules. So I think actually that the localization of the bioactive substances at the site of injury is actually critical, and that you would probably avoid systemic side effects because of that. Yeah, so TGF-beta signaling is required for alveolarization, for correct alveolarization. It's one of several known growth factors that have a role in that, PDGF, FGF, and everything else. And Wei Shi has some very interesting studies where he can do interval window perturbation of the TGF-beta system, and the timing of this is extremely critical. So that you can have short-term events occurring during development, which then have long-term consequences in the TGF-beta pathway.